So here we are sitting next to the rice field. We call it Amtoli. You have seen drone images of this kind of an endless rice field, 500 bighas or something of that order, right adjacent to my family home. And this has become the place where I'm doing all the Panchabrihi cultivation. And around me is the village, Hajipur village. We call it Kanihati because that's the ancient name for it. Recently, the new government is trying to get some irrigation water from the river. You have seen our river right adjacent to it. You have seen the drone image of the river, as well as the proximity of the river from the rice field. So, just like I described before in my birthplace, in the home, we have turned it into a laboratory for rice research, corn research, wheat research. And here in the field is the place where we are growing the rice. So we are not growing the rice in a laboratory condition. We are simply bringing the rice, individual rice plant, into the laboratory setup to count it, phenotype it properly. But the actual experiment of all the segregating population after the crosses, whether it's rice or the corn, we are doing it out in the field. So rice, corn, wheat, they all evolved in the field. They did not evolve in the lab. And what we see right now is that people are just targeting one gene and inactivating it with CRISPR-Cas9 gene editing. And they're saying this gene does that, and you have a paper published. But the point is not what one gene inactivation will do. The point is the whole combinatorial arrangement of genetic alleles, or SNPs if we want to call it, working in concert in the field condition, which determines the actual phenotype. So I think I know Jennifer Doudna discovered CRISPR, and this has just taken over to the world. CRISPR has now become a verb. Everybody is CRISPRing this, CRISPRing that. Fair enough, and in a very controlled condition, you can knock out a gene. Gene targeting, we used to call it in our time. I worked on homologous recombination a big chunk of my life. So I believe in that kind of science, hypothesis-driven research, to precisely find the effect of a gene in a phenotype by knocking it out. This is the whole basis of whole genetics. So what CRISPR is doing is reverse genetics, and where you know the particular gene, you knock it out. So this is all solid science, no doubt. But is it the science that is relevant in the field? This is where I have the question. I don't have the answer. I think in the field, when we cross different varieties, or if we do a mutagenesis, if we do crossing and then segregation, then for this region, I am measuring the phenotype, the highest number. So the performer which gives the greatest number of grain yield in this condition is what we select and we plant again. So we are doing phenotypic selection in the field. The question is, how many genes need to change? How many alleles need to shuffle in order for this situation to evolve, which is the highest yield? It's not something that you can easily mimic by doing CRISPR. How many genes you have to CRISPR in order to do that? So I think after spending a lifetime in genetics, I think the forward mutation strategy, the forward selection strategy, on a particular location is very important to find a variety which is suitable for that situation or for that location, such as our village. However, to test the role of a particular gene precisely in the phenotype, you can CRISPR it and compare it in the laboratory condition along with a control. So that is the bulk of the science. That is the science that I have spent all my life doing, and I support that science. That's a good science. But that science is not automatically relevant in developing a variety which is good for food security. In order to get a variety for food security, you have to be very compartmentalized into specific locations and develop location-specific variety with a particular weather, particular atmosphere. And this is how ancient people did. If you look into all the domestication process, varieties were selected in a particular area and was deployed in that area, and other people selected, the Chinese selected rice, we selected rice in the Bengal Delta, Brahmaputra Valley, etc. And now suddenly with the advent of molecular biology and the idea of gene knockout and looking in the effect of a particular gene by knocking it out, the whole logic has become different. The logic has become, I think it's kind of promising something that it shouldn't promise. Like we molecular biologists, I'm a pure molecular biologist, I entered biology through chemistry. And we say, look, this is our gene, like REG-D gene is the one I discovered from the scratch. No one knew about REG-D before my time. So I discovered it, and I believe that, I love REG-D, and I'm interested in the science of what happens if REG-D were to be mutated, or an analog of REG-D were to be mutated with CRISPR. So I like that, but I can't claim that REG-D is everything, or REG-D is important for biology in general. I can't claim that. Similarly, people are taking a particular phosphatase or kinase or what have you, and then they are doing CRISPR-Cas9 on it, they are knocking it out, and they are saying this is how we are going to address food security. So they are taking the specific experiment and making a broad claim. I don't think that's very useful. I think it's useful for science, but it's not useful for definitely attain a particular goal around food security. Meanwhile, we have to address food security. So I think what we should do is that we should really recruit the power of forward mutation. We should do location-specific trial. We should isolate mutants in different locations or segregants in different locations. I suggested that 87,000 villages should be converted into a center of biodiversity and crop genetics. We should not have only certain center anointed to do science. We should do science everywhere. In that spirit, by showing by example, I have started to do my science in my village. I did not go to even the Molvi Bazar, the nearby town. I did not go to Sylhet town. I certainly did not go to Dhaka, although at one point I studied in Dhaka. But right now, I brought my thinking and all my understanding of biology into this village. And through this village, I want to make progress in science.